Welcome back, everyone. Hopefully you are ready now to do some Punnett squares. So we just finished off a lot of vocab, understanding homozygous, heterozygous, dominant, recessive, genotype, phenotype, okay? So you should be very comfortable going back and forth with all of those terms. And if I say someone is a homozygous recessive or a heterozygous, what that means. But now we're going to practice doing some Punnett squares and then moving on to some other types of inheritance that don't follow all of the exact same laws um, that Mendel figured out. So he was working with the very easy system. He lucked into it, but not everything always follows all the rules or works with that dominant recessive system. So that is what we're going to talk about. So you definitely, in addition to taking your notes, you're going to need that paper or scrap paper to do some practice Punnett squares, okay? And you'll just hit pause, do your Punnett square, and then come on back and see how you did. So let's get started here. All right, so we left off talking about some of this terminology like genotype and phenotype. So now we're going to do some Punnett squares. And here we have two adorable little babies. One has the gene for melanin and one is an albino. So whether you have melanin in your skin or not, this is a simple inheritance. So if we had melanin being a big M, this baby has melanin. So she has either two big M's, right? She has the dominant trait. She could be a heterozygote. We don't know by looking at her. And then this baby is an albino. So we know that he has two little M's. Okay, so now here we've got some albino frogs that we're looking at. And I want you guys to do a Punnett square. See if you can do this. So what you're going to do is cross this black frog, who I'm telling you is homozygous dominant. Okay, so that should give you some information. What is that is their genotype. So what alleles do they have? If you're using M's, do they have two big M's or a big M and a little M or two little M's, right? That should give you information with this homozygous recessive albino. Okay, so do it once and then take the offspring that you get from that cross and cross them with each other. So those are little incestuous frogs. So go through 2 generations. I want you to pause and do that Punnett square, okay? And then come back and I'm going to solve it for you. Okay, so if we have a homozygous dominant, that means that that frog has 2 of the dominant alleles. Okay, and we are mixing that, mating it with a homozygous recessive, the white one. If it shows that white trait, if it is albino, that means that it has 2 little M's. Okay, if it shows the recessive trait, it has only recessive alleles. Okay, so we're mixing these two. So we're going to mix a big M with a little m, little m. All right, we've set this up. Now this individual can only give a big M, and this individual only gives a little m, right? So you just drop down or across whatever trait that parent is giving, okay? So these are this is our F1 generation, and they are 100% heterozygote, which means if they all have a big M, they're all going to look black, right? So their phenotype is black. These are all black frogs. Okay, now we're going to mate them with each other. So we're going to take this individual and mate it with this individual. Okay, so now we're mixing a heterozygote with a heterozygote. This is going to look different. Okay, this individual, they get a big M from this parent and a big M from this one. So what is their phenotype? Their phenotype is what they look like. This individual is black. Their genotype is what genes they have. They are homozygous because they're both the same, dominant. They have both of the dominant traits. Okay, now if we go to the top right, gets a big M from the parent on the left and a little m from the parent on the right, or from the parent on the top. So this individual is a heterozygote. is their genotype. They have two different alleles, two different genes here. And their phenotype, what they look like, is black. Okay, now we're going to do the one on the bottom left. Same as the one on the top right. This is a heterozygous individual whose phenotype is black. And then if we do the one at the bottom right, this individual is homozygous recessive, and so they're going to look white. So here we have 3/4 black. If we want the phenotype frequencies, and 1/4 white. You don't need to tell me 25% homozygous dominant, 50% heterozygous, right? I just want to know how many are black, how many are white. Give me the phenotypes, okay? 3/4 black, 1/4 white. Okay, and you can clear this and go back and do it again if that's confusing to you, okay? We're going to go through lots of examples like this. Let's go back to our slides. Okay, so we just did these frogs, and we ended up saying, remember, like 75% would be black in the F2, and 25% would be white. Well, that's the probability, right? We're working with the probabilities. If you flip a coin, the probability is that 50% of the time you'll get heads, 50% of the time you'll get tails, but you don't always actually get those numbers, right? So that's where chance comes into play. There's always a chance that things are different. If somebody has 2 kids, they don't always get exactly 1 girl and 1 boy. They could have 6 kids and end up with 6 girls or 6 boys. It's not always 50-50, right? So if we're looking at this and there's a heterozygous cross, which is what we just did, right? What was the probability of a homozygous dominant offspring? Well, we could set this up. And the probability of that homozygous dominant is 25%, right? That's this one. 1 out of 4. But you don't always get that. So if somebody has, what's the chances that if they have 3 kids, they're all going to be homozygous dominant, right? That's possible. Just because it's only a 25% chance doesn't mean that if you have 4 kids, they're going to exactly match this, the Punnett square, right? So it would be 25%, it's 1/4. Each of these are independent, times 25% times 25%. So that's actually like what's 16 times 4? 64, I think. I might be wrong on my math here. Let's see. You guys can all get out your calculators too and correct me. It's 64. I got it right. Okay. So the chances of them having 3 offspring with that same trait is 1/64. So this is just showing that both probability and chance play a role in genetics, right? Parents could have kids that all get the same trait, right? Or all of the kids could look different. So when we're talking about small numbers, you know, if you're only having 4 kids, chance is playing a really big role. It's if you had 1,000 kids, right, that things are more likely to even out. If you flip a coin 1,000 times, you're more likely to get close to 50-50 than if you only flip it 3 times. So when we're thinking about like human disease, people go to genetic counselors if they know there's they carry a disease trait in their family. There could be a 25% chance of them having that disease if you're doing the Punnett square, right? Someone who is homozygous recessive, maybe there's a 25% chance the parents could have a child with that disease. They could have 4 kids and all of them could get the disease, or they could have 4 kids and none of them get the disease. So that's where chance plays a role in genetics. Everything doesn't exactly follow that Punnett square. So that's important to understand. Okay, so we've talked now about kind of the classic cases, the Mendelian genetics. So Mendel is the monk. This is Mendelian genetics when you have a dominant trait and a recessive trait, and everything is inherited as expected. But things don't always work like that. Of course, there are exceptions. So there's something called incomplete dominance. And when things have an incomplete dominance, they blend. So this is like, going back to pre-Mendel, everybody thought things were a blend. Well, some traits actually are. So in snapdragons, which is a kind of flower, a red one crossed with a white one will actually give you a pink flower. So here we have our red flower and our white flower, and the heterozygotes, if they have a big R and a little r, are actually pink. So you may notice in the previous slide, instead of having a big and a little, when you have a big R, this implies dominance and little r implies recessive. Well, it's not actually dominant, right? You don't end up with a red, you end up with a pink. The white one has some power in here too. So sometimes for these, we'll have the color red, color white. Okay, so you can show things any way you want, whatever's easiest for you to understand. I don't want you to be confused with how things are annotated. And you can pick different letters as well. So that's incomplete dominance. There's something else called codominance. And codominance means that instead of a blend, each one still stays the same, right? Its trait shows through. So for this, if we're talking about like chicken feather color is codominant, if you mix a white chicken with a black chicken, the offspring are going to have both black feathers and white feathers. If it were incomplete instead of codominant, what would those feathers look like? So think of that for a second. So codominance, both traits show, and you get black and white. What would incomplete dominance be? Okay, so incomplete dominance, remember, is a blending. So if feather color were incomplete, you would end up with a gray chicken. The black and white would kind of blend together to make a gray. Okay, in addition to incomplete dominance and codominance, there's also the possibility that there's more than two alleles for a trait. So everything we've talked about so far has two alleles. You have a red flower or a white flower. You have a purple flower or a white flower, right? You've got two options. You've got dominant and recessive, or two different traits that could blend. That's not how things all work in the real world. There can be many alleles for a trait. So in this case, some may still be dominant, some may still be recessive, but you've got more than two options out there. Each individual can still only carry two alleles, right? You have one from your mom and one from your dad. But the population as a whole may have more than two alleles. Now, a classic example that you guys are all probably familiar with is human blood type. So there's three alleles here. There's allele A, there's allele B, and there's allele O. In addition, this is not inherited in a classic Mendelian dominant recessive way because, or it's not expressed in that way, because A and B are co-dominant. Neither one is dominant over the other. So if you get an A from one parent and a B from the other parent, you are going to have the phenotype of AB. AB is not an allele, it's two alleles. It's allele A and allele B, but you show both of them. O is recessive to A and to B. So somebody can have blood type A, and if they have blood type A, their genotype would be AA. They could be a homozygous dominant, or they could be AO. They could be a heterozygote. Someone could also have blood type B, and so their genotype for that would be, why is this not writing for me? They could be a homozygous dominant for B, right? Or they could be a heterozygote and have both B and O. Now, if they have blood type AB, the only genotype they could have is an A and a B. And if they have blood type O, the only genotype that they have is O, because O is recessive. So if you're showing the recessive trait, that means you have two copies of that. And then this just goes to show what it means. If you have blood type A, that means you have this anti-B antibody. You're going to fight any Bs that. So this is why you couldn't get a blood type B transfusion, right? If you have type B, you're going to fight type A. If you have type AB, you can get any kind of blood. You're not going to fight it. And if you have type O, you can only get type O blood. So this is a case where we have 3 alleles, 2 of them are codominant, 1 of them is recessive. But this is all one gene, okay? One gene. This is the gene for blood type. There's also, you may have heard of positive and negative. That's a different gene, okay? But this is one gene that's just A, B, and O are the 3 alleles. There's also traits that have multiple genes involved. So these are called polygenic. Poly means more than one. So it's more than one gene. So it's one trait, like height, but there's many genes involved, 180 of them for height, right? Which is why there's not just a short height and a tall height, right? It's not that everybody is either 5 feet tall or 6 feet tall. There's this huge range. So things that are polygenic and have multiple genes involved tend to have a really big range because it really depends, like all of the different alleles that you have for all of these add up together to give your final height. So there's an enormous range of heights for adults, right? For human adults, there's a really big range. So it's not a tall, short, it's a big range because there are so many genes involved. Another polygenic trait in humans is eye color. There are at least 10 different genes. So if you learned in high school biology that, like, you know, there's a dominant and a recessive, right, for eye color, you learned that wrong. If you learned that brown is recessive over blue and that is how it is, that's absolutely wrong because there's way more than 2 eye colors out there, right? There's not just brown eyes and blue eyes. There are a lot of different eye colors. There's dark brown, there's light brown, there's green, there's dark blue, light blue, right? There's a huge range of eye color and there are many genes involved. So I'm going to show you guys an example. This is a real example. So here we have on top 2 different parents, okay? And these are their eyes, and they have 2 children. Okay, so I've got 3 pictures underneath, but they have 2 children. So I want you to think about which of these 2 kids could be theirs, looking at these eyes. So pause for a second, think about it. If I label them 1, 2, and 3, I want you guys to vote, vote in your head on which 2 you think are theirs. Okay. And the answer is all of them. And I know I said just 2 kids. This individual in the middle and the right, number 2 and number 3, is actually the same person. If you look in the picture 2, their pupil's really big, and in picture 3, their pupil's small. If you've ever looked at someone who has lighter colored eyes, you may notice, depending on what they're wearing or what light they're in, their eye color looks like it's changed. Some lighter eyes, the color you're seeing is actually due to like the reflection of light around them. So this is the same individual, just in different light. And they actually do have a brown-eyed child. It is rare for lighter-eyed parents to have a brown-eyed child. But because there's so many genes involved, if one parent has a dominant trait in one gene, but recessives in the other, they may end up with a lighter colored eye. And if the other parent has, let's say this parent A has a dominant trait in gene 1, and parent B has a dominant trait in gene 3. When you have a dominant trait in both genes 1 and 3, you end up with brown eyes, but if you have it in only one or the other, you have a light color eyes. Neither of these parents is totally recessive. They don't have pale blue eyes. Pale blue eyes are recessive in every gene, as far as we know. There may be more that we don't know. But neither of these parents has that pale blue eye. So they have some dominant traits in there. So this is just throwing out everything you ever learned in high school biology. And then there is also the case, so this is one trait, your eye color, that's affected by many genes. Then there's the opposite trait where one gene can affect many things. It's the opposite. This is called pleiotropy. So in this case, and it's more of a side effect than it actually impacting all of those things. But sickle cell is the classic example here. So sickle cell is actually it's one allele, it's one base pair in the DNA. And we'll talk about this more later in the semester. But it changes your, if normal blood, let's say you've got two big H's for your hemoglobin looks normal. If somebody who is a heterozygote for sickle cell, so remember we looked at red blood cells in lab and they look like that. If somebody has sickle cell, some of their red blood cells are going to be shaped like that. It's like a sickle. If somebody is a homozygote for that, then all of their red blood cells. So somebody's heterozygote has a mix. If they have these kind of spiky red blood cells, first of all, they end up with anemia, which means they're not getting enough oxygen because this shaped red blood cell can't carry as much oxygen. And then it also pokes things. So you end up with damage to your spleen, circulatory problems. So there's all these cascading effects just from having that one gene with a mutation in it. And I think maybe we will stop here for this one, and we will pick up in a little bit with the next lecture.